Okay, let's get into it. So we're going to continue working on our microarray experiment here. We are to the point where we've put all the libraries in, and you should have downloaded the files from NASA Gene Lab for the BRIC16 experiment. So what I'm going to do, depending on if you ran this or not, make sure that you put eval equals false on these. And if you had issues loading some of these files, there is some of these packages. I'm going to add a line of code here. Let's see, where is it? Here we go. To kind of turn off the error, the connection errors. I know I had a problem with both these Arabidopsis databases. So if you add that, it is possible to kind of get around it, and that should solve your problems. So I'm going to run these libraries again quick because I started a new session, but everything should be installed already. So let's give that a second. And while that is going, we can start our next section here. Okay, so what we want to do now is those files that we downloaded from Gene Lab, we're going to start working with those. So we're going to call this cell files, a new variable, and we're going to use the function list.cellfiles. And you can see there it fills in automatically. And this is just going to look in the directory where our markdown files are. Adjust my camera a little bit here. And it's going to just get the names of everything. So if we run this, as you can see up here, we have a list, or this variable that has saved 6 names: F1, F2, F3. Those are 3 flight experiments, and then ground controls 1, 2, and 3. So that is good to go. Now we're going to get our raw data from these files. We're going to call it AFI raw because we're using an Affymetrix chip, and it's our raw data, so AFI raw. And we're going to do read cell files, and we're then going to pass it the variable cell files. So we're telling it to read in the data from our list of 6 files that we made up here. So let's run this code. And then down here, you should see it start to read those cell files. And I'm going to annotate this. Let's list and then end our data from the microarray experiment. Okay. Beautiful. All right. So let's move on here. We got all of our files loaded in. Now, one thing I want to do here quick is we have to create these targets. And so what that means is we have to kind of give a little more information to R as to what our experiment looks like and which of those files goes, which ones are ground and which ones control. Like, it's pretty intuitive for us, but R needs to be like told certainly what they are. So let me go into my working folder here. So I call these BRIC16 targets too because I remade the file for this video. But as that loads, our music playing again. What's going on here? Okay, so as we see, I've created a list, or just this really simple Excel spreadsheet or CSV file that has file name and gravity as the 2 column heads, and the file names are G1, G2, G3, F1, F2, F3, and then it breaks them into groups. So the G1s are ground, and the F1s are, or the flight ones are the F experiments. So that's it. So let's copy this exactly, save it as brick targets, brick 16 targets, or whatever, just remember the file name. So we're going to close out of that. Now we're going to load these targets into R. And so this, I'm going to just call it targets. And then we're going to say read targets, which is a command in one of our Affymetrics or lemma packages. And I'm going to call it brick 16 targets 2. So this has got to be the name of the file that you just created. Make sure it matches. Don't just copy what I'm typing if you did change it. We're going to tell it that the separations in this file, so it knows how to read it correctly, are commas. So it's a comma, CSV, comma separated. And then the row names are file name. So that was our first column. So our row names are just that, or first column, right? G1, G2, G3, etc. So we're going to run this code. Cannot open the connection. So this is an issue that multiple people have had, right? And so this could be a couple different possibilities here. A, I spelled this wrong. So I'm going to pull up my thing again here real quick. Or my folder. So what this means is that R is looking for your file and it can't find what it is because either you spelled something wrong or it's not in the right directory. So this is brick targets, brick 16 targets. See, I didn't put an underscore here. So that is a reason. Another reason is you might have your CSV file in the wrong folder. So let's try it now. Beautiful. Okay, so if we open this now, we see that we have our row names are the same as our file name. And then we have ground and flight, just like we had in the Excel spreadsheet. Okay, so let's continue on. First, let's annotate this. Let's define our targets in this experiment. Okay, and so now let's quick do a look at our data that we loaded in. So let's do hist Affy raw. So this is just a histogram of all of the data that we loaded in. And so as you can see here, we have the log intensity and then the density. So this is just showing the relative the relative intensity of certain genes, right? And so as you can see, the shape's good, all right? But what we want is we want them to overlap really well. And the reason is say you have one microarray where there's a 10-fold expression change and another one where there's a 5-fold expression change. But over that first microarray with the 10-fold change, the entire microarray is showing more increased expression compared to the second one. And so what you have to do is a background adjustment, right? And you have to normalize it. Because what can happen is on these chips, you could get too much probe added to one chip or something experimentally that causes the data to change. And so what we can do is we can control for that based on all these microarrays. So they match out much better. So what we're going to do now, oops, don't want to type right there. Let's get rid of this so it doesn't mess up our code. All right, so now let's make another code chunk. And we're going to just call this data. Bad habit, I know, but sue me. All right, so we're gonna create a variable called data read Affy. So it's going to read those Affymetrics values into its own data frame. So it might take a second. And then our next, we're going to call eset or expression set. And we're going to do AFI : RMA of that data frame. And what this is, RMA is a normalization procedure to normalize this data based on the background noise in each experiment. So let's run both of those. See background correcting, that's good. Okay, beautiful. So now what we're going to do is we're going to look at the expression again. And since we changed kind of the format, we're going to use a different, instead of the histogram, we're going to use this plot density function called of expression that we just calculated of that eset data set that we created up here. So let's run that. Expression is PRS, not RPS. Beautiful. Okay, so now if we compare these two, you see how like messy the raw data was, where we have this small peak here, this giant peak here, and kind of some middling and deviation over here. We'll now look at our normalized data here, and it's much, much better. So these experiments are much more comparable to one another. These chips are much more comparable to one another now that we did this background normalization. So we should get cleaner results. Okay. Now, we're going to add some annotations, because as I said, I think I said earlier, these, depending on what database you're getting data from, they can all have different nomenclature for their genes, right? So TAIR, which is the Arabidopsis information resource, they have TAIR IDs. And if you went to NTRES, they'd have NTRES IDs. And then some people use gene symbols if you read a paper. So like HSP in humans is heat shock protein. So that's like the symbol. So we want to kind of make a master sheet that has all these different variables in them, right? All right, so we're going to call it myFrame. And we're going to call it, make it a data frame. And we're going to do this expression of the eset. So we're just making a data frame with our eset data from before. Because if you see this, if we click on eset, it's a big matrix. There's a ton of data in here. And really, we just are worried right now about getting these expression values for each gene. And as you can see here, now this is the TAIR ID. So if you look at this and you're like, oh, look, gene 244904_AT. It doesn't mean much, so we need to annotate it, right? So let's close these guys out. So we have now our data frame with the expression values in them. And now we're going to make another data frame called annotate, and call it data frame. And then this is going to be, I have it written out in this other file here. I'm going to grab it because it's kind of long. See, this is the code that we need. So what we're doing is we're going to pull from this data frame, and we're going to use sapply. So quickly, let's look at what sapply is. So sapply, where are you going to go? Let's click on this. There we go. Come on. All right. So it's a version of lapply, and it returns a list of the same length as whatever your data frame is or X is. And then which is the result of applying this function to that element of X. So what this is doing, this sapply, is it is taking from this Arabidopsis database that we loaded, and it's taking a column, in this case gene name. Oh, don't move that. It's taking gene name and it's applying it to our data frame, and that it's pasting. So that's the function it's looking for, and then making it a CSV. And then it does the same thing for our KEGG terms, so KEGG pathways. It does the same thing for our Go, which is gene ontology, etc. So I'm going to run this. This will take a little bit because it's going through, and this big database has 22,810 observations of each one of these things. The gene name, the KEGG pathway name, the gene ontology, the gene symbol, the description of the gene, and then this ACC number, which is another kind of identifier. So we're going to let this run here real quick. But yeah, so there's a lot of, there's a package called Reshape, and there's a lot of these little tools like sapply or lapply, merge, and things like that allow you to kind of play with these data frames and kind of cut and paste, really, so you don't have to like export your file and then go into Excel and then paste everything together and then re-import your file. So you can do it all in R, and it makes your pipeline a lot easier, because say you want this pipeline to run over and over again. Say you want to save it and do multiple experiments, or microarray experiments, and just change the files you input. Well, having that part where you have to go outside of Excel or outside into Excel to like move your data around is no bueno, right? So it's best to build everything in R and have as little. You want a pipeline where you can just name your files at the beginning, hit go, and it'll do everything. So that's what we're gunning for here. So I'm going to annotate this quick. All right, get some detailed information about our genes. So then in the next section here, let's do, we're going to make a data frame called all, and we're going to merge. That was the other function that I told you, and this is to kind of combine two data frames. It says here, merge two data frames by common columns or row names, or you could use join. All right, actually, I'm going to take a peek at this annotate table since it's done running. So as you can see here now, we have our Arabidopsis number, our tear number over here. We have what kind of gene it is. So in this case, quick glitching out here. Some of these are hypothetical proteins. This is NADH dehydrogenase subunit, etc. We have a KEGG term, if it does have a KEGG term. It's gene ontology, so this tells you kind of what category the gene falls into, if it's like a molecular function or where it's located in the cell, etc. We have the common gene symbol, so this is what you would see in a paper if you read it, most likely. A description, so this says encodes a plant B subunit of mitochondria, etc. So we got a lot of information on each of these genes now, right? And then the ACC number, if that was something we were going to use in the future. Okay, so let's do all merge, and let's merge this annotation file or data frame that we just made with MyFrame. And remember, MyFrame, that was our expression data that we pulled out from the Affymetrix file, raw data. So we're going to say by X, so this means which X-axis variable you want to use, and we're going to say 0, so that is not an actual column, but just the row names. And then by Y equals 0, which would be just the column names. And then we're going to say all equals true. So we want to merge everything. We don't want to merge just like the first 5 rows or something like that. But you could if you wanted to. So it's merging these together. Actually, what we need to do is annotate this while it runs. Annotation file and our expression values. Again, this is going to take, depending on your computer speed, it might take a few seconds, because we're playing with big data here, and that's kind of what bioinformatics is about, right? So this spreadsheet has 22,000 lines and 6 columns, and we're merging it with this annotation file that has 22,000 observations of 6. So it's pasting a lot together there. All right, so now if we look at this, this is our final annotated spreadsheet. We have row names. This is all the stuff from the annotation file here. But if we go to the far right, we also have the expression value for that gene for each of our 3 flight experiments, and then for each of our 3 ground control experiments. Boom. All right, good to go. All right, so the last thing we're going to do in this video is we're going to write a table, and that table is all the data frame we just made. And we're going to call this file our brick RMA normalized and annotated.txt. And we're going to make the separation of this also we're going to make this a tab separated file. They look a little nicer if you open them in Notepad. But you could make it a CSV. You can make it a comma too if you wanted to. And then you change this to CSV. And it's writing. Okay, so now all we did here is we took this and we pretty much saved our data externally in a file that you could go into into this folder. So if I look for brick RMA, where is it? RMA normalized annotated.txt. So we just saved this file here. So if we did want to go into Excel or give it to somebody else, we'd have all of our data backed up. All right, and so that is it for this video. We got one more to go for this microarray experiment, where we're actually going to look at, we've got everything in place to look at what genes are turned on or off in space versus ground. So that's what we will conclude with in the next video. So I will see you there.